Well, in the last chapter, we talked about how DNA makes another copy of DNA. So that would be the mitosis, the S phase. But DNA works all the time. And what we're going to discuss now is how DNA can provide information, because you have about 6 feet of genetic information in every cell. And we only access a few genes per cell, so that, one might only access a couple thousand, and another one might do a completely different thousand. So when I think about how your DNA acts on a daily basis, it's more like a instruction manual, or better yet, a cookbook. You buy a huge cookbook, but you might really only do a couple of recipes. You won't really do all of the recipes. And that's how a cell is. Some cells do a few recipes, and some cells do completely different, based off of what kind of cell it is. Now, when it's time to make a protein, what we have to do is we're basically trying to figure out what's going on in the body, and we're making a protein or an enzyme or a hormone to counteract that. So what happens is your body has DNA in the nucleus. It does this thing called transcription, where it makes this sort of temporary template called RNA. Now, RNA is single-stranded, and it's allowed to leave the cell. So it's allowed to go from the nucleus to the rough ER. So it just goes to the other side of the nucleus. And then from there, that RNA becomes a template. And that template is how we can make a protein, and that's called translation. So the first one, from DNA to RNA, is called transcription. You're transcribing DNA into RNA. The next one is you're translating that RNA into a protein code. And basically what happens is you have DNA in the nucleus. It does this thing called transcription, where it makes that little mRNA or RNA. That RNA is single-stranded and gets to leave the nucleus. And then from there, translation occurs in a ribosome, and it becomes a protein. And that protein is going to be some sort of function for the cell. So what we're going to really concentrate on is going on transcription first. And then once we finish that, we'll do another video for translation. So if you recall, DNA is a long code, and it has A's and T's and C's and G's in it. Well, RNA is a 4-letter code as well, except now what happens is every time we have a, it used to bind to a T, remember, double-stranded, and if we had a C, it used to bind to a G, and this one has 3 hydrogen bonds, this one has 2 hydrogen bonds, and that's DNA. DNA. With RNA, basically what happens is A gets converted into your cell, and C still goes to G, pretend that's a G, and that's going to be bonded together. So when we see an DNA like this, what it's going to code for is an RNA sequence. So if you can, pause the video right here and fill in what you think the RNA code will be. If you can't do it, no problem. I'm going to actually do it for you. Go ahead and pause it now if you can. If you can't, or if you're done, let's see if your answer is right. G will become C. Normally A would be T if it was DNA, but now it's going to be U for uracil. T would normally be A in DNA, and it's still A here. We've got another A. Normally A would be a T, but now it's a uracil. G, U, U, C. So what's basically happening here is that every time we see a G, we're going to put a C. Every time we put an A, we're going to put a U. Just the same way we did with DNA, except the big change now is every time we see an A, we're going to put a uracil. And the uracil is something that comes apart a little quicker. and it tells the body that it is a temporary code. So now what happens is, if we're going to go from DNA to RNA, we have to do this thing called transcribing. And we have to transcribe it from DNA to RNA, and it requires a table when we finish. So first, we've already done the DNA going to RNA, just like we did in our last PowerPoint slides, where G became C and A became U, and the two T's became AA. The next thing that happens is we read DNA in a triplet code. And this triplet code, can you see how we do words of 3? Those are called codons. And codons are just like how we call words. We string a bunch of letters together and they're going to give it some meaning. Well, DNA does a code of 3. And it has just like a sentence has a starting point and it has a stopping point. So like with a sentence, you know it starts because there's going to be a capital letter. And then as you get to the end, there's going to be a period or exclamation point or something. So you know that the sentence has stopped. Well, to read these codons, what we do is we go to this table, and you'll have to look up this table and be able to use it and be able to figure out what amino acid these guys are coding for. Because these codons, when we read them in letters of 3, they each equal a different amino acid. So let's go ahead and try to do a couple. So the first one I'm going to do is AUG. And that's because it's a start codon. And I just know this, but you might have to kind of go through it. You'll notice that first thing, if we just look at the table really quickly, there's an AUG that says start, and then there's a couple that say stop. So these are going to be like our periods. This is going to be like our capital letter. It says this is where the protein starts, this is where the protein stops. And that's just because mRNA has a little bit of extra in the front and a little bit of extra in the back. And that little extra is just so it can get in the ribosome and sort of get the genetic material threaded and started. So let's go ahead and code for CUA. How we look at that is, see on this side it says first base, here it says second base, and here it says third base. Well, what that is equating to is, what is the first base? What's the second nucleotide or second base? And what's the third nucleotide or third base? So we know that our first nucleotide is C. So if you look, there's a column for U, this whole box. There's a column for C, this whole box, A, and then G. Well, we know our first letter is C, so that means it's going to be somewhere in this square. Our next one is U. So can you see how it goes U, C, A, G? So that tells me, if I kind of go like this, that my letters are going to be somewhere around here. CU, and then to finish it off, we could totally find CUA, but third base, CUA. And that tells me that my first amino acid is LEU, which stands for leucine, but on exams, if you give me LEU, you'd be fine. Let's do another one. Let's do UUC. So what you can do now is pause the video and you look up UUC, and then I'll start up and we'll do it together. Okay, well, let's do U. So remember how U is the first base? This one's pretty easy because U is the second base as well. So that's going to be right over here. And if we look in this box, UUC, which means that it is phenylalanine. Cool. So when we read RNA, it's going to be very similar. RNA to DNA. It has a different sugar. That's really the only big difference. It's got ribose instead of deoxyribose. It's got a nitrogenous base, which are those A's and C's and G. And remember how now it has U for uracil? And then it's got a phosphate. Unlike DNA, it's single-stranded, And there's 3 types that we're going to talk about, but there's actually like 22 different types. We're going to talk about what an mRNA is, a tRNA, and a ribosome, or rRNA. This is basically the big difference, is the sugar. The sugar is a little difference in both of them, and that's what's the main difference. The next thing is that RNA is usually single-stranded, and DNA is double-stranded. And also, DNA has a thymine, and RNA has a uracil. In the next video, we'll talk about the 3 types of RNA.